What has been your proudest accomplishment in the SIOD lab?
I think probably the telomere identification, but I think that I also think that it's just been great to get a group of people together to sort of be our research team, and I think that's something that's a work in progress, and we're continuing to expand, but I think that we're now well on our way to having a really great research team. And the other thing I'm proud of is that there have been a number of scientists at Stanford and elsewhere that I've been able to tap for their expertise at no cost to the foundation or to me, but they've just been very generous with their time. And we're hoping that we can have these people as actual collaborators on the research projects that are, for example, at some point funded by the National Institutes of Health. But you have to lay a lot of groundwork and network with people and talk about the science first. And I think we have a really good relationship with a number of other scientists. So I'm very optimistic that we're going to move things forward now quickly.
What do you foresee happening in the next 2 years with SIOD research?
I think we're going to, at minimum, be able to test our hypothesis that our main hypothesis is that this protein, that DNA replication is not sort of an even thing. So the way to kind of envision it is that a cell has only 5 hours to replicate its entire genome. That means all the chromosomes. And it does it by having multiple sites of replication scattered, like hundreds per chromosome. And then the results of this replication are stitched together into an intact chromosome. And we think that depending on the particular region of DNA and its particular structure, that some of these are trickier and are more dependent on SMARCA1 than others. So we predict that these regions will vary from SMARCA1 deficiency compared to healthy individuals, and that those regions that are difficult to replicate, what will happen is, in the absence of SMARCA1, their structure will change so that they can't function normally, and that we will find genes that are kind of located within these regions that account for why the T cells function is affected, why the kidney cell, the podocyte, is affected, why the cartilage cells are affected, and so on, and the vascular cells. And the idea is that if we can figure out the mechanism by which that gene function is being shut down, we may be able to kind of bypass that problem using possibly drugs that may already be out there, or if necessary, we can come up with new drugs. But we really think that what we're really hoping to do in the next 2 years is show that there's a common disease mechanism that we can kind of go after for treatment and possibly cure. And that's what I'm hoping to do in the next 2 years.